So good morning, everyone. Today, I'm very glad to introduce some of my research here. My topic is the molecular analysis of microbial hotspots in rhizosphere use correlative chemical imaging analysis. So here, what's a microbial hotspot? This is a figure from the review paper. The hotspots are small soil volumes with much higher process rate and intensive interactions compared to average soil conditions. Here we can see that most of the soil, they just have a very slow process rate. They even called dead soil, but a small amount of soil they called hotspots. We can see the relative volume in hotspots is about 1% to 10%. But his processing rate can be 10 to 100 times faster. So here is a schematic illustration of 4 microbial hotspots that grows in soil. This is also from this review paper. We can see they just have 4 typical hotspots. The first one is a detritosphere, how to spell it, detritosphere. It's just the litter on the soil interface here. So that's definitely, we believe that's a hotspot. Another famous hotspot is the rhizosphere. And the third one is the biopores, especially with, we can see this with earthworm or some inside in earth animals. And then that's if there's even that's no active worms, that this channel that still can regard it as a hotspots. So PGPB in rhizosphere, as I showed, the rhizosphere is just a well-known microbial hotspot. And some of the microbial in rhizosphere are plant growth promoting bacteria as PGPB. However, rhizosphere interaction are a complex function of a bio and abiotic factors, making it difficult to directly translate the result from the lab result to connect to the field environment. So the need is therefore detailed molecular and elemental understanding of plant microbial interactions in the rhizosphere is very important for us to fully understand and harness in the future. It's engineer its hotspots to help the bioenergy and agriculture applications. So why correlative molecular imaging is needed? The first reason is rhizosphere is actually a highly heterogeneous system. We know that the root is, we know the microbial, we know that the mineral particles and organic matters, they stay in a system and it's not homogeneous distributed. However, most of commonly used chemical analysis tools in this field, such as total carbon analysis, FT-ICRMS, NMR, and genomics, and some other proteomics, this kind of analysis tools, they are mostly the bulk analysis techniques. They are lacking of spatial resolution. So another had good spatial resolution advanced imaging tools such as the fluorescence scanning electron microscope, transmission electronscope, light source. They can provide very vivid imaging. However, they cannot provide the molecular information. And also they are mostly not surface sensitive. So time of flight secondary ion spectrometry is a powerful surface analysis technique. The basic principle is we use 10 to 30 keV primary ion beam to sputter the surface to eject the secondary particles out. And then we can use acceleration voltage. Normally, most secondary ions, they are only single charged. So they, in the same field, they will get the same energy. If we allow of these ions to fly a certain distance to get to the detector, because they have the same energy, but they may have different mass. So they have different speed. They will get to the detector at a different time. So based on time of flight, we can know exactly mass to charge ratio, and we can get the mass spectrum from surface. And of course, we can do scanning of the beam to the surface to collect the spectra from different locations. So we can do the chemical imaging. So TOFSIMS is interesting because it can provide a decent spatial resolution. And how decent? Normally XY resolution can go to sub-micron. Some good situation, we can even go to 0.1 micron. And we can also do 3D imaging in the Z resolution is pretty good, is a few nanometers. Molecular recognition, here we can see in the principle, when we sputter the surface, some clusters can be ejected out directly. So small molecules, they can be detected directly. So they say that the molecular recognition capability. And the third one is it's a very surface sensitive technique. The only about 1 to 2 nanometers materials will be sputtered away from the surface. Of course, if we use 3D imaging, we need to do sputter layer by layer. Although TOFSIMS is powerful, however, we know each technique has its own advantages, but also have some limitations. So to understand the complex system such as the rhizosphere, correlative imaging definitely is needed. Here is a strategy we want to use in our research. In this work, we developed a sample holder for fluorescence, TOFSIMS, XPS, and SEM imaging of the same location, exactly same location of the samples. So fluorescence, why we want to use fluorescence? Because as a fluorescence has been widely used for the attached on the PGPB, so we can know the distribution of the PGPB. And then if we go to the same location, we use TOFSIMS, we can get the imaging, use imaging to make it sure we go to exactly the same location. And then we can collect the spectrum from this location to get the chemical molecular information. And also we can use XPS imaging to find exactly the same location and do high resolution XPS spectra to know the chemical state information of the sample surface. Of course, it's a SEM, scanning electron microscope, can provide a very decent spatial resolution. It can go to the 10 nanometer, 20 nanometer spatial resolution. It can provide much more vivid topography information. So this one is also needed when we want to explain data. So this is the basic strategy we want to use in our research. Here, brachydodium sample preparation. When we want to study the brachydodium, is a very interesting model system. It's a model grass. It's a very important and interesting bioenergy crops because it can grow in the marginal land very well. So here we just grow brachydodium in the soil, and we have reference with no PGPB, and we can put some PGPB, for example, Pseudomonas, use this way. And then we can also add some other PGPB, just the Astrobacter or other species. And how can we immobilize the sample? Is after brachydodium grow to a desirable stage, we can pull it out, and we can cut the leaf, we can cut the stem, we can cut the root. The root, we need some gentle rinsing to remove the soil first. And we can immobilize the sample holder to do the imaging analysis. Here, definitely in this research, we focus more on the root. Here are some details about the sample holder and the sample mounting. We designed this special sample holder. It looks like not complex, but it still needs some thinking. The first, it has a desirable size. The size is 50 millimeter diameter and with a 6 millimeter thickness. Why we want to use this size? Because this size is compatible with Amazon's quite a few imaging techniques, including fluorescence, time of flight SIMS, nano SIMS, XPS, and SEM. At least there is 5 commonly used tools. And another detail here is we can see we prepare a trench outside. So to make it sure all the screws is lower, slightly lower than the top. So this is very important because there's some microscope technique, for example, fluorescence or top sims, nanosims, they have very short working distance. So if we want to detect analyze this root sample, we need to make it sure that the sample is right height and compatible to the short working distance for the different microscopes. Here is some details shows the brachipodium root mounting. After mounting, we can see here we have some labeling mentioned I1, I2, U1, U2. Actually, this is 4 plants. In each plant, we can we use the 2 root samples from each plant. And here we can see we use a specially designed mole window. This is have a 3 stripe window here, so we can track each root 3 locations. And this kind of window, why we design of this? Because this is a very easily for our track for different locations. For example, okay, I go to this location for fluorescence, and then when I move to another one, for example, top sims, we can move to this location very easily to find exactly same location to do imaging. So here we can see that we have 4 plants, and altogether 24 locations can be easily tracked for analysis. Here is a very interesting. I just mentioned that our sample is U1, U3, I1, I2. This is 4 plants. So what's the difference among them? Is the U1 and U3 actually reference samples? They grow without Pseudomonas PJPB added. And I1 and I2, they grow with Pseudomonas PJPB. So at our original expectation, we expect for the I1, I2, altogether 12 locations, we can find Pseudomonas easily. However, the result is surprising. Only 2 locations, these 2 locations. Here, we observed Pseudomonas. Suppose we altogether have 12 locations from the Pseudomonas modified plant root. So this result suggests that the Pseudomonas is not easily attached on the root surface. So the PGPB, although they can promote the plant growth, they may not be promoted by directly interact with the root surface. And another situation is a question arises here. So if there's only these 2 locations, they have fluorescent, they have Pseudomonas. So what's the chemical difference between these 2 locations and other locations? So then we need some other chemical analysis tools, for example, top sims. So we can collect the top sim spectra from 24 interesting locations. Here is some representative top sim spectra. Left side is negative ion spectrum, right side is positive ion spectrum. Here we can see we can labeling each peak. And we can see among samples, the spectrum looks similar. They have some difference, but how can we distill the difference out with some mimic visual, like we can visualize the difference? That's challenging. So normally in this way, there's some advanced analysis technique is needed and try to visualize the difference among the spectrum. So how can we do that? Is the PCA analysis. As a principal component analysis has been widely used in the mass spectrometry analysis, and it can distill the difference out vividly. So here's one example of the negative ion spectrum. Here we have a PC1 scores. They're very interesting. Most of the samples have the PC1 scores between the 0.1 to minus 0.1. However, only 2 spots is far away from the major groups. As our check, these 2 spots is just corresponding to these 2 spots. That is the Pseudomonas enriched area. So that is to say all Pseudomonas-free locations have similar chemical environment. But what's the difference, chemical difference among the Pseudomonas-free and the Pseudomonas-attached area? We need to go to the PC1 loadings. In PC1 loadings, the positive loading is oxygen Cn and phosphor-related species. The negative loading is chlorine and sulfate SOX related species. Based on the PC1 scores, the Pseudomonas area has a relatively low score. So that is to say the Pseudomonas location has more negative PC species, has more SOX and chloride, but less Cn phosphor-related species than Pseudomonas-free locations. Here, if we go to the positive ion spectrum, we can get the similar result. Most Pseudomonas-free area, they have shared the similar PC1 scores, and only these two locations have a relative positive PC1 scores. So that is to say that's similar with the negative one. Also, the Pseudomonas-free locations, they have similar chemical environment, but that's Pseudomonas area, they have more Pseudomonas have more sodium but less ammonium, potassium, and nitrogen organics than the Pseudomonas-free locations. So if we can also check the exactly same location use XPS. Here is some representative XPS data. We can see XPS data show that Pseudomonas locations have more sulfur but less nitrogen, potassium, and silicon than Pseudomonas-free locations. And these locations are actually qualitatively consistent with the TOF-SIMS TOF-SIMS data. So here is SEM images of after the TOF-SIMS measurement. So this one, the left side one, SEM image shows the I2 plant at location 1. This is the Pseudomonas attached area. We can see it's relatively smooth. However, as a comparison, this is a Pseudomonas free route. They looks like a lot of particle attached on the surface. So we think this is we believe this is soil mineral particles. So here is a schematic illustration of the behavior of Pseudomonas on the Brachypodia root surface. Here, when Pseudomonas bacteria come to the Brachypodia root surface, there's only weak interactions between the bacteria and the root. So most bacteria will repulse away and go to somewhere else. Here we can also see there's some relatively strong interaction between soil mineral particles with root. However, only at the same time, yeah, okay. Here is only a small amount of Pseudomonas can take the soil particles off and attach on the root surface to form biofilms. It can take only a small amount of bacteria can form EPS biofilm on the surface and take the particles away. So this is our data suggests our correlative technique is very interesting and will be very potentially used in the rhizosphere research. We have some further study about the plant microbial action at Brachypodia seed surface published a few months ago, but today I don't have time to discuss this. So finally, I'm working in the environmental molecular science laboratory, and the work was supported by the EMSL. So we have a collection of state-of-art instrumentations, and we also have world-famous experts. So both academic and industry collaborators are very welcome. So thank you for your attention. If any questions, I'm very glad to answer.